it's really it's really honor and then I feel really honored and it's really my pleasure to introduce today's speaker, especially the speaker were Chen in similar in the same institution where I did my postdoc. I now like want to introduce her to today's audience so that the audience will know her better. Um, okay, so today's speaker, Dr. Meng Wu, Assistant Professor of Molecular Microbiology at Washington University, School of Medicine in St. Louis. Dr. Wu received her PhD in human and statistical genetics from Washington University in Jeff Golden's lab, following her undergraduate studies in biological and biomedical sciences at Tsinghua University in Beijing, China. She completed postdoc training as a research fellow at Harvard Medical School and then later served as a research scientist at Philips Research North America. Dr. Wu leads an innovative research program at the intersection of microbiology, immunology, and system biology. Her laboratory focuses on deciphering the complex interactions among the microbiota, stroma cells, and the immune system, particularly the complement system in the gut and its role in host defense and disease modulation. In addition to probing the microbiota stroma cell axis, her work spans multiple interorgan systems, including the gut-bladder, gut-brain, and gut-memory gland axis, with the aim of uncovering novel therapeutic targets. Dr. Wu's groundbreaking work has been recognized with the prestigious NIH Innovator Award from NIAID in 2024, and her research has been published in top-tier journals such as Cell, Nature, and Science. So today, it's really exciting to have her to give us a seminar. Please join me in welcoming Dr. Meng Wu, whose research is shaping the future of microbiome informed precision medicine. Please, please, Dr.
Well,
Wu.
thanks. Thank you so much for the really generous introduction, Paul, and thank you so much for the organizers' kind invitation. It's truly an honor to be part of this really exciting seminar series. Today, I'll be focused on the gut complement system, recently identified local complement network in the intestine. I'll discuss about the interactions between this gut complement system to commensals and pathogens, also share some our latest findings in this area. So I will really briefly talk about the gut microbiota and its function. So as many of you probably already familiar with, there's trillions of the microbes living in the gut, including bacteria, fungi, and virus. There have been numerous studies in the past decades to show the critical role of the gut microbiota in host health. Broadly, their function can be grouped into three categories, including resistant to the colonization by pathogens, transfer of the ingested compounds, including food or the medications, and also the development and maturation of the immune system and the nervous system. Those functions are not mutually exclusive to each other. Today, I'm going to tell you about how the gut commensal microbes can modulate the immune system to defend against pathogens or some local cells. So this really inspired by this when we study the interactions between the host microbiota, a striking observation is different organs actually respond to the microbes different. For instance, when a single microbes come to the circulation system in the blood, we would tend to kill them right away. However, when the commensals come to the gut, the gut actually welcomes them and even let them stay with us. We carry billions of them every day with us. However, if a pathogen come, then the gut have to turn on their killer mode again and kill those pathogens. So how this process actually get regulated is what I'm interested in. And such a process is really critical for human health because any improper response to these events can cause disease. For instance, if we couldn't kill the microbes in the blood system right away, it can cause sepsis. And if we actually overreact to the commensals, it causes inflammation. And if we couldn't kill the enteric pathogens, that can cause diarrhea or any other disease. So I will briefly talk about how we kill the bacteria in the blood. The complement system was discovered over 100 years ago by Nobel laureate in the 1890s, and it gets the name as complement because it can pose more than 15 different proteins, and they can enhance the antimicrobial potency of antibodies. In other words, complement. So that's how they get their name. And when the microbes come into their blood, the complement cascade gets activated and kills bacteria. And here's a more like detailed how the complement cascade get activation and the function. So they majorly activate through 3 different pathways: the classic pathway, the lectin pathway, and the alternative pathway. All these 3 pathways merge into the central component C3. Once C3 get activated, it cleaves into 2 different forms. One is C3A and one is C3B. So the C3B can bind to the bacterial surface, and this process called C3 opsonization. After C3B bind to the bacteria, there are 2 ways to kill bacteria. One is to recruit other proteins like C5 to C9 and form a membrane attacked complex on the surface of the bacteria. The other one is to recruit phagocytosis, such as macrophage or neutrophil, and they will engulf the bacteria and then kill the bacteria. For the past 100 of years, we have been learned a lot about the complement system, but they always consider as a hepatocyte-derived and serum-redundant system. Recently, there are more studies have revealed the local complement system, such as in the lung. Then for me, we are really interested in the gut, so we have this question as, does the complement system exist in the gut? On one side, it sounds possible because as we know that the gut have a most densely populated immune network, probably because they also have the most density microbes. There's a famous saying, talk about 70% of your immune system lives in your gut, so it's possible complement might be there as well. However, on the other side, we know in the blood, they kill the microbes right away with so many microbes around. If complement system exists, how they actually make peace with these commensals? So we start to do the literature search first, and we find there really, really very little is known about the complement system in the gut. There are some reports, but mostly in IBD patients or IBD models. As you may know that in IBD, the gut barrier function get breached, so there might be some blood in the lumen, so that could be a source of complement. Little is known about if the complement system actually exists under homeostatic environment. Something boosts our confidence that it might exist is when we search for the public available database, we find that there's C3 gene expression in colon tissues from like mice studies. So that's suggesting there's a local expression of C3. With this knowledge, we decided to do a systematic investigation of the complement system and also trying to answer the question how they interact with commensals and pathogens. So today I'm going to talk to you about 3 questions. First, we want to answer, does the complement C3 exist in the gut, as that's the central component of the complement system? Then, is it induced by the commensals? If so, can gut complement C3 still find against enteric pathogens? And how about commensals? The third question we want to answer is, what types of cells in the gut produce complement components? So when we started this question, we're really trying to use our favorite system, the germ-free mice and the SPF mice, to answer the question. Germ-free mice are the mice, they are raised in the bubbles. So for their entire living environment, they're never exposed to any microbes, including bacteria, fungus, or virus. We first use ELISA to measure the C3 protein levels in the fecal samples from germ-free mice or SPF mice. We find that there's a substantial amount of protein C3 in the SPF mice fecal samples, but not that much in germ-free mice. when we look into the serum C3 levels, we find there's not significant difference. So this suggesting there's a local C3 protein in the fecal samples from SPF mice, and there seems locally response to the microbiota colonization as the serum C3 level is still very similar to each other. To further answer the question if this is a really microbiota causal relationship or just a correlation, we took two different approach to answer this question. First, we can colonize a germ-free mice with SPF feces, so that after 2 weeks, the germ-free mice carry the microbiota from the SPF mice, and then we take their fecal samples to measure C3 protein. And we also can take the SPF mice, use the broad spectrum antibiotics to treat it and examine if there are any still live bacteria in the gut. After 2 weeks of treatment, we couldn't detect any live bacteria in the gut, and when we collected the fecal samples, we could examine their C3 levels. So we find that after 2 weeks of treatment of antibiotics, the SPF mice fecal samples have a C3 level as low as the germ-free mice. And for the germ-free mice colonized SPF microbiota, they actually have a C3 levels as high as SPF mice. So this suggesting that the secretion of the luminal C3 is inducible and also reversible. Then from many human studies, we already know for humans, there are different microbiota composition there. So then the question is really being if there are any if the C3 levels are actually different because of different microbiota. So we take advantage that many studies have already shown that for the 3 biggest vendors in the United States, Jackson, Taconic, and Charles River, even for their black 6 mice, they have quite a different microbiota. So we want to use them as a model to answer this question. And we got the mice from those different vendors. So they are all black 6. We took 8-week-old mice. We did the 16S sequencing just to examine if their microbiota are different. And you can see their microbiota are quite different. And then we measure their C3 levels in the fecal samples. We find that the Jackson mice have quite low C3 levels, while the Charles River mice have much, much higher, actually 10-fold higher than the Jackson mice, while Taconic mice have C3 levels in between. This is really interesting to us, suggesting that the microbiota composition could be a way to regulate the luminal C3 levels. But if it's actually a causal relationship there, we did the microbiota transplantation. So we colonized germ-free mice with the fecal samples from Jackson or Taconic or Charles River. And we find all the recipient mice really recapitulate the C3 levels compared to their donors. So you can see that the germ-free mice colonized with Jackson microbiota have a C3 levels as low as Jackson, while the germ-free mice colonized Charles River microbiota have a C3 levels as high as Charles River. And it's almost exact same levels with their donor. So this really established that the luminal C3 levels are transmissible by the microbiota. So next we think then that's suggesting it might we might be able to actually modulate the microbiota to modulate luminal C3 levels. But how can we do that? We first need to test if this experiment all done in germ-free mice, but if we actually will be able to change SPF mice microbiota and then change their C3 levels. So to answer that question, we first did a co-housing experiment as the mice are coprophage. So just by co-housing them together laterally, they will have a microbiota transfer. We co-house Jackson and Charles River mice, and after 2 weeks of co-housing, we collect their fecal samples and then measure the C3 levels. We find that the Charles River mice co-housed with Jackson don't really change their C3 levels that much. There's a little bit of increase in there. But for the Jackson mice co-housed with Charles River, their C3 levels increased significantly compared to their controls. So that's the same Jackson mice in the facility for another 2 weeks. With this, we think that the Charles River microbiota probably carry some really high C3 inducers. To find those high C3 inducers, when we measure the microbiota composition between the Jackson mice or the co-house mice or the Charles River mice, you can see they are really, really quite different. So to do that, we actually, the Dennis Lab developed method, we do a short period of co-housing to kind of have a limited transfer of the microbiota. Then we can sequence the microbiota to identify the difference. So we co-house the Jackson mice to Charles River mice only for 1 day, then separate them, then collect fecal samples 2 weeks after and examine their fecal C3 levels. We find that even just exposed to Charles River mice for 1 day, the Jackson co-house mice have a C3 level as high as Charles River mice. And then we did the sequencing of the microbiota from the Jackson mice and Jackson co-house with Charles River for 1 day to identify if there are any microbial species difference. And we find that we identified there are 3 species that are transferred into the Charles River to the Jackson mice, significantly more abundant compared to the Jackson mice. And the Protena is the top hit, and their abundance and percentage is consistently high among all the recipient mice. So with this result, we decided to further testing if we actually be able to introduce one single species to change the composition of the microbiota and then change the C3 levels. So I isolated Protena species from the Charles River mice, and we also have a control, the Proteus mirabilis as a negative control, as we know that this species only exists in Charles River mice, but not show up in the 16S analysis I did. We also actually use fecal ciliary from Charles River mice as a positive control to see their effects on C3 levels. We colonized those mice for 2 weeks, and then we examined the C3 level in the fecal samples. So you can see that for the Jackson mice, we did check that the Proteus mirabilis was successful in colonized Jackson mice, but they didn't really induce a significantly higher C3 levels. While the Protena species introduced to Jackson mice, it could significantly increase the luminal C3 levels. And the Jackson gavaged with Charles River microbiota have even higher C3 levels, probably due to the additional species we showed that also can correlate with C3 levels. So with this really suggesting even one single species will be able to change the C3 levels in a more complex microbiota settings. Then we are really interested to know if this kind of phenotype is also true in humans. So collaborate with Dr. Ansel Xiao's group, we will be able to get healthy human cohorts fecal samples and perform the human C3 ELISA. From this healthy human cohort, we defined that in healthy humans, there's there's C3 protein in the fecal samples, and they are quite different. Some people have as low as like 30 nanograms of fecal samples, and some people have as high as 700 nanogram per gram of feces, C3 in their feces. So the next question is this also transmissible to the germ-free mice? So we picked 2 donors. One is donor 1, which have a low C3 levels, and another is donor 8, which have a high C3 levels. Then we transfer their microbiota into germ-free mice, and after 2 weeks, we measure the C3 levels from the fecal samples of those notobiotic mice. We find that the germ-free mice colonized with donor 1 microbiota have a much lower C3 levels compared to the germ-free mice colonized with donor 8 microbiota. So from this result, it is suggesting first, the human-derived microbes also can induce C3 protein in the notobiotic mice. And then the second is the resulting C3 levels really recapture their donor C3 levels, suggesting that notobiotic mice could use as a model to study the relationship between the microbiota to the C3 production from human samples. In summary, I just show you that first, C3 is detectable in fecal samples from healthy humans and mice. And then these luminal C3 levels are microbiota dependent. We have been identifying the Protena species as one of the high C3 inducers. Once we discovered those, we are really interested to know what this complement C3's function in the gut. We started from the traditional complement function, the infectious disease, and we choose the mouse model of infection with C. reduction. The reason we choose C. reduction is first, it's a non-invasive gram-negative enteric pathogens, means they are not really going to the blood to just kind of distinguish between the blood complement and the local complement. We know their pathogenesis mechanism is forming the attaching effacing lesions in the intestinal epithelium, and that mimics the human pathogen EHEC and EPEC. So we start from using winning stage mice, and we find that in the C3 deficient mice, when they infected by C. reduction, they actually die. There's 50% of the mice die after 20 days of infection, while the white type mice survive through the entire course. And then we move on to the adult mice, and we find that the C3 deficient mice have a significant weight loss compared to the white type mice. Well, those C3 deficient mice are the global lookout. As we are investing in local complement here, how we can actually rule out the serum C3 function. So to do that, we use a protein that called a copper factor, which have protein structure very similar to C3b. As I told you, when C3 function in the blood, we know that the C3b can amplify the alternative pathway and activate C3 to let it degrade. So by inject CVF by IP, we will be able to specifically deplete serum C3. You can see after 1 day of injection, the serum C3 level dropped very low, and also day 4, but the fecal C3 levels still maintain the same. So now we will be able to use this white type mice treated with copper factor to deplete serum C3, but still have the gut C3 existing there. And we did an infection with C. reduction, and you can see that the white type mice treated with copper factor really have a weight loss phenotype more similar to the white type, suggesting that the remaining fecal C3 in the gut could do the protection. And when we check the pathogen burden, we also see that for the C3 deficient, the global C3 deficient mice, the pathogen burden is much higher compared to the white type, while the white type treated with copper factor behave more like a white type mice here. So those results together suggesting that the local C3 could be sufficient to protect against the enteric pathogen. Then we really want to take a look, where are those pathogens? So collaborate with Yuan from Jeff Moffitt's lab, we will be able to visualize that. And you can see in a C3 deficient mice, this C. reduction, they are located in the epithelial layer, while you look at the white type mice 7 days post-infection, they are actually not in the epithelial. And if you did a normalization by the epithelial length, you can see a significant difference there in the C3 deficient mice compared to the white type. While we examine the C3 proteins in the fecal samples along the infection, we observed a significant increase of the C3 protein, suggesting that the host is actively upregulate C3 expression and also production in the gut. So we want to see if C3 actually activated working against the pathogen. So using a GFP positive C. reduction, we will be able to try to detect C3 deposition of the opsonization on the pathogens. So you can see that after 7 days post-infection, when we gated on the GFP positive C. reduction, we can detect the C3 deposition in the white type mice, but not the isotype control or the C3 deficient mice. And this is a statistical showing it's quite significant. So C3 is just one component of the entire complement system. How about other complement proteins? I'm going to just summarize what we're finding here, because that involves a lot of different experiments we done. So first, we actually use C1q deficient mice to examine if the classic pathways are required, and we find that in C1q deficient mice, C3 deposition still happen to C. reduction, suggesting that's not required. And then we use MBL deficient mice to examine the function of the lectin pathway. And in the gut, it's also not required for the C3 deposit to the pathogen. We don't have a alternative pathway deficient mice, but we examine the factor B upregulate gene expression, and we find that the factor B upregulation significantly during infection, suggesting in the gut it probably works through the alternative pathway. And then we are wondering how about downstream pathways. So we first trying to examine the protein C5, as we know it played a critical role in the formation of the membrane attack complex, but we couldn't detect any fecal C5 protein under homeostatic environment or even during infection. And you can see the C3 protein level actually increased significantly during infection. So we couldn't detect any C5 during infection, kind of suggesting probably not really existing there. And we also did an RNA-seq for the colon tissues, and we also couldn't really detect any gene expression of C5 to C9 during infection. So all this suggesting the C5, C9 probably not really existing in the local gut complement system. Then we are looking for the phagocytes. So we examine the neutrophils or the macrophages, and we find the neutrophils were recruited to the colon significantly during infection, and then also use the GFP labeled C retention, we will be able to show that C retention was engulfed by the neutrophils during infection. So with all this, we think that in the gut, the C3 works through the alternative pathway, and then for downstream, it works through the phagocytosis pathway, and the C5 to C9 not existing in the gut. Then this really come back to our original question that how the gut complement system deal with commensals. With knowing how gut complement system work, we think that because C5, C9 is not existing, so they are not MAC formation in the gut, and under homeostatic environment, since there's not much phagocytes actually around, so they won't be able to, even C3 might deposit to commensals, they won't be able to actively clear the commensals under homeostatic environment. To test that, we examine the composition of the microbiota in the C3 deficient mice compared to their wild type nitimated control, and we find that there's no significant difference in terms of the composition of the microbiota, either based on the presence of species, that's the alpha diversity showing here, or the different abundance, that's the beta diversity showing here. So with all this, we think that the gut complement system can defend the pathogens when they need it, but they are not really doing much to the commensals under homeostatic environment. And remember, I told you that the gut commensals actually can modulate the C3 levels, and we know that different people have different C3, different microbiota, and we also frequently hear that different people have different susceptibility to infectious disease. So we really want to move on to test the hypothesis that the interpersonal difference of luminal C3 level could be a risk factor for infectious disease. To avoid any potential genetic drift between Jackson and Charles River mice, we actually took the microbiota from Jackson and Charles River, colonized the germ-free mice, and then examined the C3 levels. We showed that the germ-free mice colonized Jackson or Charles River microbiota did cause a different fecal C3 levels. We then infect those mice with a much higher dose of C retention, as I showed you before. We find that for the mice colonized with Jackson microbiota, they have a significant weight loss compared to the germ-free mice colonized with Charles River microbiota. And when we examine the pathogen node, we also find there's much more pathogens the C retention in germ-free mice colonized with Jackson microbiota. So this gives us conclusion that the microbiota probably can colonize the gut, cause a different luminal C3 levels, then the lower C3 levels, the infection severity increase. In summary, in the second part, I'm just showing you that the luminal C3 is sufficient for protection for pathogen infection, but not actively clear commensal organisms under homeostatic environment. And that in the gut, the C3 is actively activated through the alternative pathway and functions through neutrophil phagocytosis during infection. Then the nominal C3 levels, which is modulated by the microbiota, can correlate with the host susceptibility to the infection. With this, we are really excited, but there's a really fundamental question we feel like we don't know here is what types of the C3 cells in the gut produce C3. To answer that questions, we collaborate. Dr. Claudia Kemper from NIH provided a C3 reporter mice to us, and using this reporter mice, we will be able to unbiasedly to examine what kind of cells produce C3. So we sought all the C3 positive cells from the gut and performed a single cell RNA-seq. From the result, we find that there's 3 major cell clusters in the gut producing C3. And using canonical markers, we will be able to identify what they are. So we find that the green cells here are the epithelial cells. They represent around like 9% of the entire C3 expressing cells in the gut. And then we also have the myeloid cells, which is around like 12%. Then we have a huge population of cells produce C3 as the stromal cells. They are almost like 80% of the entire C3 expression cell population under homeostatic environment. So we were really surprised and also curious, what are those stromal cells? So stromal cells, by definition, are the structure cells that provide essential connective tissue, stroma, and usually we consider they define the architectures of tissue by deposit extracellular matrix. Well, before we move on, we just feel like we needed to be cautious about this because this is a report mice, maybe something just weird. So we decided to actually take a Y-type black 6 mice and then sort out these different populations, the epithelial cells, CD45 cells, and then stromal cells, performed a bulk RNA-seq to see if this is also holds true in Y-type black 6 mice. And we did find that the IECs have a very low C3 level, and the CD45 have a higher compared to C3, and stromal cells have the highest C3 level expression. So that's confirmed that it's not just the reporter mice, it's in the black 6 mice as well. And we also examined the human single cell RNA-seq data from the gut cell atlas from Tiedemann lab, and we pull out all the complement genes, and the x-axis here showing the different cell types in the gut. If we take a look into the C3, you can see majority of the C3 expressed in the mesenchymal stromal cells. So this is very consistent with our mice data. And then for other genes, for C5 to C9, you can see they really express a very low level in the gut, except a little bit C7. And then for C1q, A to C, they are high expressed in myeloid cells. That's also consistent with the mice data we get. So all this data showing that for the gut complement system, seems the gene expression pattern are very consistent between the human and the mouse, which suggests that this might be a conserved system during evolution. So now we really want to know where are those stromal cells, and collaborate with Shika and Daping, we will be able to do a immunofluorescent staining on the reporter mice, and we find that those red cells, they are the T tomato, in other words, C3 expression cells, and we use PDPN as a marker for the stromal cells. So you can see there are stromal cells out there, but the majority of C3 expression stromal cells are located in this conical lymphoid follicles. Those are the structure considered as tertiary lymphoid structures, and they're studied before showing they are induced by the microbial colonization, and they are also the site have a lot of immune cells. So this is really interesting to us that the stromal cells expressing C3 located in this very special structure. Again, we want to confirm it's not just a reporter mice. So collaborate with Sai Yuhang in Genentech, we will be able to perform the C3 iron scope study using the Y-type black 6 mice, and you can see majority of these C3 expression cells located in the conical lymphoid follicles, and they are just the C3 gene expression probe. So with this, we've been able to confirm that the majority of those stroma cells primarily located in the chronicle lymphoid follicles. And since we use two different approach and two slightly different genetic background of mice, and they are in two different facilities, one in the East Coast, one in the West Coast, we really consider this is a robust phenotype. So the next question we want to answer is, does stroma cells really be able to actively respond to the microbial signals? We developed an ex vivo stroma cell culture system to test that. So we'll be able to isolate stroma cells from these C3TD tomato report mice and then culture them in the lab. Then we will be able to add fixed bacteria or molecules to test if the stroma cells can respond to that stimulant and express C3. So you can see that we first use a fixed entire microbiota to test it. There's a increase of TD tomato signal in those cells. And then when we measure the protein levels in the supplementant, we also detect much higher C3 from the cells stimulated with fixed bacteria compared to the controls. Then we want to know if any individual bacterial species can do it. So we will be able to find that compared to the controls, B. fragilis can do it, Protena also can do it, while the E. faecanis and the E. fischem couldn't do it. So this is from the TD tomato signal, and we also did the protein level test. We did find that for our test, the C3 protein levels in the supplementant is quite consistent with the C3 expression level measured with TD tomato. So really confirm us the TD tomato signal can be used to represent the C3 protein production. So we want to understand why there are different bacterial species behave differently. So we turn back to our single cell RNA-seq data, and we find that for those C3 expressions stroma cells, they express high levels of TLR4, but not that much of LOT2, which suggesting that they may use those different receptors to respond to the microbial stimulate differently. And as we know, the TLR4 is LPS receptor, and the LOT2 is MDP receptor. So we use this two different molecules to do the stimulation, and we the result validate our hypothesis. LPS can stimulate the C3 production in a high level, while the MDP couldn't do that. And we actually give a much higher levels of MDP compared to the LPS, really test if the difference is real. After that, we want to see if the C3 response we observed from those in vitro culture actually also holds true in vivo. So we use the infection model again that we were previously, I showed you that we observed the C3 protein level increase significantly during infection. Now with the reporter mice, we will be able to measure how the C3 expression changed locally. So we're showing here that the C3 positive cells among the entire live cells. You can see that for all the cells I showed you before, the epithelial cells, the CD45 cells, and the stroma cell together, they increase significantly during infection. And then if we zoom into stroma cells, if we look into the gene expression levels of C3 in stroma cells, you can see they increase significantly during infection. And just 5 days post-infection, you already see the difference. And if you see the percentage of the C3 positive stroma cells among all the stroma cells, you can also see an increase just 5 days post-infection, and that's consistent to the 14 days post-infection, which consider the peak of infection. So in summary, we will be able to use different technologies such as single-cell RNA-seq and also sorted cell bulk RNA-seq, immunofluorescence, RNA-scope, ex vivo culture system, and also infection model all together to show that the C3 cells located in the intestinal lymphoid follicles can sense the microbes and express C3. And we believe this is the first time that showing that stroma cells can participate in the immune response against infection by produce C3. In summary, our study really showed that in health populations, there's C3 protein in the fecal samples from both mice and humans. And then the variation in the microbiota can cause a different luminal C3 levels, and these luminal C3 levels negatively correlated with the severity of infectious disease. We will be able to further identify the cell type that produce the local C3 and identify that the stroma cells located in the isolated lymphoid follicles are one of the dominant populations to do that. During infection, that because the neutrophils and other phagocytes come in, then the C3 deposition to the pathogens will help that a complement-mediated phagocytosis process, and also the host increase their C3 expression levels. Together, we really showed that the intestinal complement can fine-tune the immune response against pathogens, where they still can safeguard the gut's reddened commensal organisms. So this work largely being done during my postdoc study in Dennis Kasper lab, working with Wen Zhen and also Xinyang Song, and also other collaborators. Their contribution that I have been acknowledged along the slides. Currently, since launching my lab 2 years ago, we've been really focused on exploring the dynamics between the microbiota stroma cells and the immune system. A major focus have been on the complement system I just talked about. So we are looking for different infectious disease models in the gut, and also we're expanding our work to other mucosal sites, such as urinary tract. In addition to infectious disease, we are also really wondering this kind of interactions happen in other disease processes, such as inflammation or cancer. So this is my team here, and we are really grateful. We get supported by the NIH New Innovator Award besides my staff, and please feel free to reach out if you have any questions I don't get to answer in the next few minutes, or if you are interested in joining us or exploring potential collaborations. My contact information is here. Thank you so much for your attention.
Okay, that's